Hey everybody, I hope you had a great weekend and ready to start the week off. Unfortunately, I'm not going to be here today, but everything is set up and you are all prepped and ready to be able to knock out this lab. So, our goal today is to have this meiosis bead lab done today in class. So, to kind of get us started here, I'm just going to open up our module 4. We're going to be in our last module, module 4.4 meiosis. Now, there's two links here that I want us to get us started on. Here's the chapter 4 textbook. If you open that up, I'll do this with you here. This lines out everything in chapter 4 in the book. Feel free to grab a book for this lab if you want a physical book for this, but I went ahead and scanned everything that we'll need. And so the page that you want to go to is page 138, and this is just going to lay out very beautifully and go into good detail what is happening each stage of meiosis. So please make sure to kind of follow along with this as we go along with this. But let's go ahead and open up our activity for today. The meiosis bead lab should be here, should be ready to go. All right. And so, what you and a group of 3 are going to do is you are going to take what we learned on Friday, talked about how we go from a diploid germ cell to 4 haploid uh, sex cells. And so, you're going to apply that in a model format using the meiosis pop beads. All right. So, our goal is, again, use the beads to represent what is happening to the DNA as it goes through cell division. So pretty straightforward here. And when you go back to the lab, you're going to find the tables all set up with the red and white pop beads. Or I'm sorry, red and yellow pop beads ready to go. All right. So there's a skeleton outline of the stages of meiosis. So you're going to use the pop beads to show what is happening to the DNA as it goes through. And so let me show you an example here. So you'll see in one part of the table, you'll have it in interphase. And so in this particular picture right here, we see we're starting off with 2 chromosomes, all right? The yellow representing the chromosome inherited from your father, the red chromosome inherited from your mother. And so this is going to be interphase. This is going to be in chromatin form, all right? Make sure you kind of understand what stage and what form the chromosomes are taking. And then when they go into prophase, obviously you can see that they have replicated and they are in their sister chromatid format here. Now, I want to take note in prophase 1, this is where crossing over is taking place. So make sure you are indicating the synapses showing the crossing over taking place and be able to show me how that goes through as you are following the pop beads. And as you can see, there is, we end up having 2 chromosomes with metaphase. Then again, this is the law of independent assortment in which the homologous chromosomes line up independently with one another, and then you're going to work it through. So by the end, you should be able to tell me exactly how each of these gametes are going to be haploid and all genetically different at the end. Okay. So, and then there is a practice that I'll want you to do as well. They'll be set up in your module 4. If you have any questions, please email me, and you all have a great day.